Good afternoon, ladies and gentlemen. Today Professor Hennings will explain how to estimate the African slave ancestry of white Americans and the European colonial ancestry of black Americans.
That is correct, Karen. The New World has been a mixing bowl since Columbus's time, and today we shall see how to measure the extent of the genetic mixing in the United States.
I am looking forward to it.
The New World was colonized during the Ice Ages. When the ice melted 16,000 years ago, sea level rose, genetically isolating the New World from the Old. Then, for thousands of years, diseases swept the Old World, forcing its inhabitants to develop immunities. Native Americans never developed similar immunities. Within 2 generations of Columbus's landing, 95% of the Native American population was wiped out by Old World diseases to which Europeans, Asians, and Africans were immune. Some people refer to this misfortune as genocide. But it was not intentional. The newcomers had no idea that their mere presence would kill so many innocents. Over the next few centuries, the decimated population of the New World was replenished by millions of people from the Old, carried across the Atlantic in sailing ships. 11 million came from Africa. Most of these were taken to the New World against their will as slaves, although some became indentured. 3 million people came from Europe. Most came as indentured laborers, although some became slaves. Incidentally, the difference between indenture and slavery was that indentured forced labor was for 7 years and not hereditary, while slavery was lifelong and hereditary. To historians of the New World, of Earth's many human populations, 3 large groups are of special interest. First, those populations who were already in the New World when Europeans and Africans arrived. Second, those who came from Europe. And third, those who came from Africa. Autosomal DNA markers measure the average continent of ancestry admixture in your genome. The technique was devised for New World inhabitants to measure what fraction of their ancestry came from each of the 3 demographic sources, Africa, Europe, and Native America. The 2 scientists who pioneered autosomal DNA mapping are Mark Shriver of Penn State University, on left, and Rick Kittles, now at the University of Chicago, on right. Dr. Kittles was with Howard University at the time of the original research. This is one of the scatter diagrams that they made after measuring admixture in several hundred subjects. Each dot represents an individual. Each is plotted left to right depending on the ratio of European versus African admixture. People of 100% European DNA are on the left edge, those of 100% African DNA are on the right, and everyone else is proportionally in between. This particular chart was meant to see if admixture matched skin tone, so each dot was plotted higher or lower, representing darker or paler skin. The trend is upwards, and to the right. This shows that, although you can see hundreds of exceptions on the chart, in general the more African admixture you have, the darker your skin tone. Of interest to the historian is that each subject was also asked his or her racial self-identity. Those who considered themselves white were plotted with tiny diamond shapes. Self-identified African Americans were plotted as tiny circles. With that in mind, you can see that self-identified white Americans ranged from 0 to 25% African admixture inherited from slave ancestors. On the other hand, African Americans span the entire range. And there is a lot of overlap in the range from 15 to 25% African admixture. This graph is from a different study. It also matches the ancestral African versus European DNA admixture of several hundred individuals with their ethnic self-identity. Dots are plotted with subjects of 100% European admixture at the top, and individuals of 100% African admixture at the bottom. They are plotted in 4 groups. From left to right, the groups are 147 white Americans, 264 African Americans, 135 inhabitants of Zaire, formerly Congo, and 159 inhabitants of Nigeria. As in the prior chart, there is much overlap between white and black Americans in the range of 0 to 30% African admixture. Some white Americans have over 20% African genetic admixture, and some black Americans have little or none. Here are two important things to notice about the two leftmost clusters. First, the black and white groups are not symmetrical. The mean African admixture among white Americans is about 0.7% African and 99.3% European. Only about 1/3 of white Americans have detectable African admixture, averaging about 2.1%, but this varies regionally, from 0 in the north to 5% in the Cumberland Plateau. The second important point is that virtually all black Americans have some European admixture. They average about 83% African and 17% European admixture. Again, this varies regionally, from 25% European in Philadelphia, to 0 among the Geechee Gullah people of the Sea Islands of South Carolina and Georgia. Interestingly, the United States is the only nation in the Western Hemisphere with two clusters, two genetically distinct populations. Every other country in the hemisphere has a single scattering of dots centered on the admixture ratio that matches its colonial populations of Europeans and Africans. Argentina's cluster would be at the top of the chart, Haiti's at the bottom, and Puerto Rico's in the middle. There is sometimes a mismatch between a person's autosomal DNA markers and his or her external appearance. Only about a dozen genes determine visible traits, such as skin tone, hair texture, features, compared to hundreds of markers that track ancestral colonial source populations. Since heredity at each generation is random between parents, it is not unknown for people to inherit European appearance while carrying African markers or vice versa. For example, the man on the left has about 11% African DNA admixture, and the man on the right has nearly 30% European. Why is there such a discrepancy between measured genetic admixture and physical appearance? There is an immediate answer to this question, and a deeper answer. The immediate answer is that many different invisible genes identify continent of ancestry. As of the summer of 2010, the HapMap project has identified hundreds of single nucleotide polymorphisms, markers, that reflect a person's ancestral continents of origin. On the other hand, only a handful of genes encode for the few superficial, externally visible features, such as skin color, hair curliness, etc., that Americans see as racially significant. Parental genes are randomly recombined with each passing generation. And so, it can happen through sheer chance that an individual like Dr. Shriver can inherit many invisible African DNA markers, but few or none of the handful of gene variants that encode for African racial appearance. Alternatively, a person like Dr. Kittles can inherit those few gene variants that encode for visible African appearance, but otherwise inherit many invisible, but ancestrally informative European markers. The deeper answer becomes evident if you plot each of the two U.S. endogamous groups from the skin tone diagram separately. This graph shows just one portion of the population of the earlier chart, those who self-identified as African Americans. You can see a positive correlation, R-squared of 0.211, between skin tone and African admixture. On average, if you are considered an African American, the more African admixture you have, the higher your melanin index. There are many exceptions, of course, like Dr. Kittles for instance, but on average this is the case. What is more important is that the range of skin reflectance as measured by the melanin index of African Americans is very large, spanning a vertical distance of 50 points from 30 to 80. African Americans span a huge range of skin tones. This graph shows the other US endogamous group from the earlier chart, those who self-identify as white. In this diagram, you can see that there is no correlation, either way, R2 equals 0.001, between skin tone and African admixture. Clearly, if you are considered a white American, your melanin index is unrelated to your degree of African admixture. More importantly, the range of skin reflectance as measured by the melanin index of white Americans is very narrow. It spans a vertical distance of only 20 points from 20 to 40. This is less than half of the range of skin tone variation found among Black Americans. The combination of narrow phenotype variation, skin tone, along with a wide range of genotype variation, Afro-European admixture, among white Americans, shows that a selection process has taken place. Few human populations display such a clear mark of selection. Narrow phenotype variation alone does not necessarily indicate selection. Northern Europeans display little skin tone variation, but they lack a wide range of African admixture. Broad genotype variation alone does not necessarily indicate selection. Dominicans average 50/50 Afro-European admixture, but they also display a wide range of skin tones. The late Stephen Jay Gould, Harvard biology professor and columnist for Natural History magazine, explained this principle with a baseball analogy. Plot a scatter diagram of the batting averages of 1,000 amateur or minor league players and you will find a very large range of variation. A few such players are bad, a few are good, but most spread across the entire range in between. Now plot the batting averages of professional athletes in the major leagues. All are very good indeed. More importantly, the range of batting averages among them is tiny. The difference between an outstanding star of the game and a rookie is a matter of mere hundredths of a percentage point. The reason, of course, is because you cannot get into the majors unless you are very good at it. Similarly, wild cows vary greatly in the amount of milk that they produce. Cows in a dairy farm produce more milk on average, but also their milk production varies very little among themselves. Those cows who do not make the cut become hamburger. And so, why do few, if any white Americans display a strongly African appearance, have a high melanin index, despite having detectable African admixture? Because those Americans who look black are assigned involuntarily to the black endogamous group, whatever their genetic admixture. The scatter diagrams are not symmetrical, because the selection process acts only upon the white group. As revealed in court records, discussed in other sessions, a person of mixed ancestry who looks European like Dr. Shriver, in practice has the option of either adopting a white self-identity or a black self-identity. But a person of mixed ancestry who looks African lacks such a choice. U.S. society assigns such a person to membership in the black endogamous group, like it or not. U.S. society has unwittingly applied selection pressure to the color line. The only American families accepted into the white endogamous group have been those whose African admixture just happened not to include the half-dozen gene variants for dark skin, or the other physical traits associated with race. Since those particular gene variants were sifted out of the portion of the white population that originated in biracial families, the relative percentage of the remaining, invisible, African gene variants in this population cannot affect skin tone. In conclusion, the very fact that skin tone does not vary with African genetic admixture among white Americans, despite their recent African admixture, confirms that physical appearance has been an important endogamous group membership criterion throughout U.S. history. It has resulted in genetic selection of the white U.S. population for a European racial appearance, regardless of their underlying continent of ancestry admixture ratio. And, with that, I conclude today's presentation. Thank you for listening.
Thank you, Professor Hemmings. Well, that is our time for today, ladies and gentlemen. This is Karen Sharp.
And Randolph Hemmings.
Signing off until next time.